complex 23, which is, as we all know, is historically associated with colonization. We could observe that also the semen strains presented a lower titer when compared to the rectovaginal isolates. And taking the sequence type 144, which among our sampling was the most common, we could also observe that the strains follow the same pattern, with the semen isolates also presenting lower geometric mean titers when compared to rectovaginal isolates. So then we started to think why that might be happening, why the semen strains are presenting reduced susceptibility to opsonophagocytic killing. And as a first step, we are investigating the C3b deposition through the measurement of C3c. So for that, we labeled the bacteria with the hottest stain. So we could, as you can see in this first graph, isolate the bacteria population of interest. And then after setting this first gate, we set the second gate on the isolated bacteria to conduct further analysis. These plots on the right and on the bottom are we used an anti-C3c antibody conjugated with FIT-C to measure the fluorescence. And these plots are to show the difference in the peaks, showing that on top here, we have the peaks more to the left, and on the bottom, we have the peaks more to the right, showing that when we add the sera, we have a shift in peak, having more fluorescence intensity when we have the antibodies. So doing that, we could observe that for the rectovaginal isolates, we could not observe a proportional correlation between the mean fluorescence intensity of C3c and the geometric mean titers in the OPKA. But on the other hand, for the rectovaginal isolates, we could observe a clear correlation. So that meaning that the more C3b is deposited on the pathogen surface, we have a higher geometric mean titer. They are killed more by this assay. And in the end, when comparing both sources of isolation, the MFI of both sources of isolation, we could also see that the semen strains presented a lower fluorescence intensity when compared to the rectovaginal isolates. And that corroborates with our results from the OPKA, showing that indeed, for these isolates, these semen isolates, C3b deposition on the pathogen surface is in fact associated with less C3b deposition on the pathogen surface, associated with higher resistance or lower susceptibility to opsonophagocytic killing. So until now, we could observe that regardless of the source of isolation, most strains belong to serotype 1, 8, and 5. All of isolates were susceptible to killing using SHRS of the matched serotype, but with a large range of titers. Semen isolates had lower geometric mean titers when compared to rectovaginal titers. Semen isolates belong to serotypes 1, 8, 3, and 5 had lower GM titers when compared to rectovaginal titers. Clonal complex 23 and sequence type 144 semen isolates had lower geometric mean titers when compared to rectovaginal isolates from the same clonal complex. Complement deposition was lower among semen isolates when compared to rectovaginal isolates. And complement deposition was associated with opsonophagocytic healing only among the semen isolates, but not the rectovaginal isolates. So I would like to thank both of my families from my lab in Rio and now my new family in the UKHSA, the pathogen immunology group. Of course, all of our collaborators and the funding agencies. And that's it. Thank you.
Thank you very much. We now have our first Q&A. So if I could invite the speakers again, please, to the front.
Oh.
So questions to the speakers?
I have a question for Kelly. I was wondering if you were able to look at the different serotypes of GBS. I mean, we've been talking about this quite a lot this morning. So
Yeah.
if there is more association with Candida with some serotypes rather than other ones.
Yeah, great question. If I'd been able to adjust my talk, I would have added that bit in. But we've looked at various invasive isolates across different serotypes. We have a cohort of isolates from pregnant women in the US, and they all aggregate or bind to Candida. There is some variation, but there was no like significant correlation that suggested a certain serotype would be better at interacting. So yeah, good question.
The Candida had to be in the hyphae form, does it, to associate?
We've been able to test that. So we have strains that are either yeast locked or hypho locked. And GBS can bind to both, but it does prefer the hypho. So I think, and that makes sense in the way the biofilms are structured, that it's going to bind the hypho preferentially, but it still can bind the yeast form. So that answered your question. Yeah.
Okay, my question is the intrapartum antibiotic. When do you propose to give tetracycline? In that model that you showed, it cannot be given beyond 4 months, right?
No. I mean, that is just the resistance genes to the class of antibiotics. So antibiotics, when you give them, and the resistance genes associated are often not linked to that antibiotic. So we've got like non-cognate resistance genes can happen. So yeah, so no tetracyclines being given to babies. It's just
Well,
look at
to the
the
mom
resist-
also.
Or the mom also, no. Yeah, so it's just because any antibiotic exposure can increase the number of resistance genes and often to like not aligned classes. So we looked at all of them, but the tetracyclines were not significant.
Thanks.
Any more?
My question to you was, when looking at other strains, you only showed 1A, 2. We did talk about all the other strains.
Yeah, we're now looking at the five serotypes that we found on the whole genome sequencing, which is 1A, 1B, 2, 3, and 5. But we now have some strains of serotype 4, and we isolated the first colonization strains that belonged to serotype 9. But they didn't go to whole genome sequencing yet, at least. They were only confirmed by latex agglutination, but we'll then proceed to look at those other serotypes.
My other question probably sounds silly, but we did a lot of work looking at men and vaccine. So is there an idea that men will need a vaccine too in the future?
Yeah, that's what we're trying to see, like, because as I said, we are conducting this research right now to try and see if there's a sexual transmission of GBS. So we're not sure that actually we'll need a vaccine for men, but maybe they could carry some strains that could be transmitted to women or vice versa. And giving a GBS maternal vaccine for the women could prevent that transmission to men. Exactly.
Any more?
Question to you about the resistance gene. So, how does it happen that there are resistance genes to antibiotics that were not given to different antibiotics?
Oh yeah, so this is, it seems like, I was very surprised when I first realized, because I'd always thought, you will give the antibiotic, the bacteria, or it will promote the bacteria that has resistance to that antibiotic, and then you'll see that resistance. But often in like epidemiological studies, any antibiotic exposure is often exposed, associated with this high ARG prevalence that is sometimes unrelated to that. And that was what we were trying to get at over here, that you're not just seeing penicillin binding protein increasing. You're seeing all kinds of resistance genes, but it is related to more the compositional change, because when it's selected for Klebsiella commonly has lots of antibiotic resistance genes. So then even antibiotic resistance genes that are not related to penicillin go up because that has selected for that taxa. And why I'm saying this is important is because if it's very related, then you just have to think about like antibiotic cycling, right, to kind of prevent. But if it's related to taxa selection, then you can think about other things like probiotics, to kind of get rid of that compositional difference if you have no other choice, like vaccination, which we may have, and then it would be a redundant topic. But
Thank you.
Thank you very much.
To our speakers and to the audience, a round of applause. Have your seats. So the second session are the flash talks. These are 5-minute talks. So first speaker is Anne Clarice.
Hello everyone, my name is Ana Clarice Mexage. First, I would like to thank the organizer for the opportunity to present my work at this conference. Today I will briefly talk about fluoroquinolone-resistant group B Streptococcus in Brazil, evidence of expansion of serotype 5-1690 sublineage with unique traits. As we know, GBS is important to cause neonatal infections and cause infections in no pregnancy adult. It's a relevant veterinary pathogen because it can cause bovine mastitis. And currently, GBS is a concern and emergent microbiome resistance threat. GBS is included the World Health Organization list in moderate medium group with reduced to susceptibility to penalizing, and classified by CDC as a pathogen with moderate concern to resistant to clindamycin. In this study, we analyzed human isolations from our collections. We evaluated the capsula type, ST, genes of resistance and virulence, and the mutations using GBS type pipeline. Now I will talk about the results regarding fluoroquinolone resistance distribution. We identified 4.1% of our collections. 13 had mutations in both GRA and GPAR-C, 5 had mutations only in PAR-C, and 1 had mutations only in GRA. Regarding of the years of resistance, is 2009, the rest from 2050 onward. It is important to understand the distribution at this mutation. We can see capsula types and ST among fluoroquinolone resistant is worth mentioning. One fluoroquinolone resistance was identified as serotype 1A ST103. This ST is generalist. In Brazil, it is isolated in both human and cattle. It's important to mention it too. Semi-isolated show the wider frequency at this profile, and in this group, serotype 5-1690 was the most frequency and was also identified in rectovaginal clinical isolate. This profile accounted for 90% and showed mutations in both GRA and PARC and showed rare leg fluoxetine mix. So, fluoroquinolone resistant serotype 5 acetyl-90 stands out. Most common profile shared by semen and rectovaginal clinical isolated showed double mutations and rare mix. But is serotype 5 acetyl-90 common in our collections? This profile most commonly found in semen isolates, and I'm happy to share more information about semen isolates when finished the sessions. And this profile increasing occurrence in rectovaginal isolated. In addition, 6% of all serotype 5 acetyl-90 in Brazil are fluoroquinolone resistant. This information leads us to think, is fluoroquinolone resistant acetyl-90 different from susceptible ones? These results answer that questions. We can see two clades. The first clade represents fluoroquinolone susceptibility, and another clade represents fluoroquinolone resistance, represented by color yellow. Fluoroquinolone resistant differed and carry ALP1, and forms a distinct clade in the phylogenetic tree, suggesting they represent a separate sublineage. In addition, fluoroquinolone resistant serotype 5 acetyl-90 had multiple resistance genes. So these results bring conclusions or more questions. What I can say that these fluoroquinolone resistant at a higher frequency in semen isolates and showed unique genetic traits. Thank you, everyone.
For this, we do it a bit differently. Any questions for her? Questions?
You said the MIC is increased, yeah? The minimum inhibitory concentration is increased, yeah, for this. By how much? Double or 10 times? Or?
In semen, actually, this profile is most commonly associated with resistance, and the most common, but in rectovaginal increase.
Okay, okay, thanks. So it's more resistant in semen.
Yes, yes.
Oh, thanks.
Thank you. Any other? Thank you. Our second presenter, Victoria.
Good afternoon, everyone. My name is Victoria, and I'm a natural products researcher. So I look into nature, maybe plants, animals, microorganisms, marine organisms, or even mineral elements for solutions into human problems, including GBS and bacteria. So, I don't want to go into what have been said since morning about GBS. A lot has been said about it. And what caught my fancy about it is that the virulent factors of the bacteria, which I mean the molecule or the structure of the bacteria that contributes or that enhances its ability to cause diseases, be it in mothers or in neonates.